Adderall is a prescription medication approved by the FDA for use in patients with ADHD and narcolepsy. The drug promotes increased ability to focus and acts as a stimulant for the central nervous system, allowing those affected with ADHD to perform better in school and in the workplace. Adderall is a mixture of 2 salts of the amphetamine stereoisomers dextroamphetamine and levoamphetamine. As an amphetamine derivative, Adderall can be both addictive and dangerous for users. Other amphetamines include methamphetamine and ecstasy, which are also highly addictive. All amphetamines, including Adderall, pose risks to the user's health, such as sudden death, heart problems, and even psychosis, manic behavior, and aggression. The FDA calls for careful evaluation of the risks posed by a patient's history of illness before starting treatment with Adderall in children and adults. As a Class B drug in the U.S., consumption is considered illegal unless prescribed by a physician. The growing epidemic of recreational use of Adderall in spite of the law is particularly concerning given its composition and potency. 13% of college students report misuse of drugs in this family in their first year, and 31% report misuse by their 4th year. Most students use the drug to enhance their academic performance and their ability to stay up late studying. Misuse of the drug, particularly if users consume other drugs or alcohol, can be disastrous. Addiction to Adderall or reduced ability to function without it is also a major concern. Amphetamines like Adderall are thought to affect monoamine levels in the central nervous system. MRI studies suggest that long-term treatment with amphetamine decreases abnormalities in brain structure and function found in subjects with ADHD, and improves function in several parts of the brain, such as the right caudate nucleus of the basal ganglia and the striatum. Adderall operates through 3 primary mechanisms to presynaptically increase the concentration of monoamines such as norepinephrine and dopamine in the synaptic cleft. The first way that amphetamines increase monoamine production is by competitively binding to TAAR1 in the presynaptic cell. TAAR1 and its homologs use 35 residues to form pockets to attach to ligands. This receptor binds to monoamines and molecules that resemble them. TAAR1 is a 7-transmembrane domain GPCR whose activation results in increased levels of CAMP through activation of adenylyl cyclase. Increased levels of CAMP lead to the increased production of monoamines to be released into the synaptic cleft. The second mechanism works through VMAT2, a single amino acid chain of 514 residues, which transports monoamines from cytosol of the presynaptic neuron to synaptic vesicles. Amphetamines bind at VMAT sites to alter its function. VMAT2 is essential in the presynaptic neuron's ability to facilitate the release of neurotransmitters into the synaptic cleft. The third main mechanism by which amphetamines increase the synaptic concentration of monoamines is through the dopamine transporter DAT. DAT stops the action of dopamine by causing its reuptake back into the presynaptic neurons. The reuptake mechanism is as follows. 1 dopamine molecule or 2 sodium ions initially bind to the transporter protein, followed by binding of 1 chloride ion to the transporter. The inwardly directed sodium current provides energy for an inward movement of dopamine against its concentration gradient. Adderall is thought to cause a reversal of dopamine transport through DAT while also blocking future dopamine reuptake. Let's investigate the structure of DAT further. The protein database code for human DAT is 4M48, discovered by Aravind Penmatsa and colleagues at Oregon Health and Science University using Drosophila. This is the structure of the protein, with side chains in green, nitrogen in blue, and oxygens in red. If we look at the structure in cartoon view, you can see the motifs that make up the protein. To make this easier, we will color the protein by chain. There are 3 chains: chain A, which is composed primarily of alpha helices, and the heavy and light chains comprised of beta sheets. Chain A is the transporter chain. An example of a binding site on chain A from residues 40 through 50 is shown here in red. We will make the chain more transparent so you can see it better. The dopamine transporter has an N-terminal tail containing 60 amino acids. Amphetamine has been shown to increase kinase activity, and N-terminal phosphorylation of DAT may play a critical role in the dopamine overload caused by amphetamines. It's hypothesized that phosphorylation of at least 1 N-terminus serine residue is necessary for amphetamine to flood the synapse with dopamine through the dopamine transporter. Amphetamines could increase the level of DAT phosphorylation. A mutation of the 5 N-terminal serines to alanines, preventing phosphorylation, inhibits the action of the transporter, indicating that amphetamine phosphorylation action may take place on these serines shown here. We hope that from this video you have learned more about the mechanisms of action by which Adderall produces its effects. In the description of this video there are links to the sources we used so you can learn more. Thank you.